data on this topic and hearing some input from your attendees. So I do represent sort of a team of researchers, not only my own lab, but a very expansive group at the University of Michigan who does adrenal research. And we're also part of a larger consortium that's been called A5, which is an American, Australian, Asian Adrenal Alliance of researchers that bind together on some studies that relate to rare diseases of the adrenal. So today I'm going to discuss a little bit about adrenal physiology because that sort of sets the stage with regard to the pathology of adrenal disease, and then move to the pathologic aspects of aldosterone. So for those who don't think a good deal about aldosterone, it's not always been around. And from an evolution standpoint, aldosterone production started with the transition of vertebrate animal species from water to land. Glucocorticoids occurred much earlier in vertebrates, all the way back to hagfish and lampreys. But aldosterone itself appeared with tetrapods, so that when we look at amphibians, reptiles, birds, and mammals, we all make aldosterone. And it's certainly thought that the appearance of this hormone evolutionarily was important for the regulation of sodium and water retention, which was important because of short supply on land. Mammals control aldosterone very tightly through the renin-angiotensin-aldosterone system, where we have, as with regard to sodium, we have a drop of sodium in the distal tubules leading to renin production, renin leading to angiotensin 2 increases, and that will feed back and cause the adrenal glands to produce aldosterone and carry us back to an increase in renal sodium retention. So it's a nice tight feedback loop. My laboratory has spent a great deal of time looking at the signaling cascades within adrenal cells that regulate aldosterone production. And one of those we've characterized is the role of angiotensin 2, the major physiologic hormonal regulator, and the pathways used by angiotensin 2 within the cell. And these tend to converge, whether it's angiotensin 2 or extracellular potassium, on calcium signaling pathways within the cell. And these calcium signaling pathways activate transcription factors that lead to increased transcription and expression of the protein aldosterone synthase. So aldosterone synthase, and I'm going to be calling it CYP11B2, that's the cytochrome P450 nomenclature, is almost solely expressed in the adrenal, and it really determines the capacity of the adrenal itself to make aldosterone. Now, reaching back into evolution, we can test the survival advantage of aldosterone by putting animals on a low or deficient sodium diet. And here we've done that with rats, where we put them on a high sodium diet, a normal chow diet that you feed rats, also a sodium deficient diet. And this was done by visiting fellow Koshiro Nishimoto in my laboratory. And what Koshiro found is when he looked at plasma aldosterone levels, he saw that even within short periods of sodium deficient diet, that we start to see an increase in aldosterone in the circulation, which is greatly amplified as time goes on, and this is 72 hours of diet. And if we look within the adrenal for transcriptional changes, we see that that enzyme I mentioned, the transcripts for aldosterone synthase, are already going up within 13 hours and even much higher in sodium deficiency for 72. But what you can also see is that under high sodium conditions, and that's really more relevant to what we are exposed to, that we start to suppress this enzyme and we suppress aldosterone production. We've done full transcriptome analysis of all the genes that change in the adrenal under sodium deficiency and high sodium diets. And this is the sodium deficiency group here. And once again, the enzyme transcript is one of the major changes that we see within the adrenal. And that enzyme, again, will carry out the final reactions from deoxycorticosterone to aldosterone, again, controlling the capacity of the adrenal to make this steroid hormone. The adrenal itself starts to remodel under sodium deficiency. This is high sodium, 13 and 72 hours, normal sodium here. and deficient sodium. And what we see that 13 hours, we don't see a lot of remodeling occur, but by 72 hours, we start to see this zona glomerulosa, the zone responsible for aldosterone production, significantly increase in size, again, helping to produce more aldosterone so these animals can survive and maintain some sort of blood pressure under this very deficient sodium diet. Now, this translates well to humans, and this is an interesting studies that were done in the 1970s, actually out of groups that were at the University of Michigan. A geneticist, James O'Neill, shown here. O'Neill's group, in collaboration with endocrinologists, demonstrated that a group of native South Americans, the Yanomamo tribes who live in this region of South America, live on effectively extremely deficient, or as they wrote a book about it, without salt, where the sodium intake hovers around 50 milligrams per day. And again, most of us are ingesting upwards to 4,000. And in this, these tribal peoples, the aldosterone is extremely high, 150 nanograms per deciliter around that area, renin extremely high, and that's what's driving this aldosterone and helping the natives to actually maintain a normal blood pressure. However, salt is a tasty component to diet, and many cultures over the decades have used salt trading as a way to, as a commodity for trading, whether it's in North America, Asia, or in Africa. And certainly now we have ready access to sodium, and it would be said to be one of the most highly available spices and used in foods, leading to an extremely high sodium diet with a suppressed aldosterone and a suppressed renin being the result. Now, from a physiology standpoint, the aldosterone was critical, again, from an evolution from water to land, but this is a good segue for us to think of the new sodium diets leading to a pathologic aldosterone state. So to compensate for our higher sodium chloride intake, most of us are easily compensate by suppressing our aldosterone and our renin levels. However, elevated sodium diets that we're all accustomed to have the potential of amplifying negative effects of even small increases in autonomous, so I'm going to be using the word autonomous some for this, but this is just renin-independent aldosterone production and other diseases of aldosterone excess. So from a historic standpoint, I think of these as sort of the big three named adrenal diseases. Certainly adrenal insufficiency, which was described initially by Thomas Addison in the 1800s, Cushing disease and syndrome described by Harvey Cushing in Boston in the 30s. And then finally, the topic of today, primary aldosteronism, described again here at the University of Michigan in the 1950s. And as these overview prevalence points across the point below indicate, certainly of these diseases, primary aldosteronism will be the most common of the three. Now, there are multiple subtypes to primary aldosterone, which I'm going to define today as a renin-independent aldosterone production from the adrenal glands. And it's important to remember that the impact of primary aldosteronism is greater on cardiovascular disease outcomes than a sex and age matched patients with essential aldosterone, essential hypertension. So the outcomes with PA are going to be much worse. Now, subtype wise, the most common cause of PA is bilateral idiopathic hyperaldosteronism, where both adrenal glands are involved in the disease. And that's followed by aldosterone-producing adenomas. And in our case, adenomas is much more commonly used than the word tumor for these particular tumors. And finally, there are some rarer cases, including familial subtypes of primary aldosteronism, which represent between 3 and 5% of primary aldosteronism. Now, over the last 10 years, There's been a lot of excitement in our particular field of the adrenal field because there's a series of mutations that have been defined. And these can be germline, so familial mutations, or they can be somatic within tumor mutations. And these are the different genes that have been described that currently cause excess aldosterone production. And there are mutations in everything from potassium channels to calcium transporters, chloride channels, and calcium channels themselves that are activating mutations. All of them seem to converge on the pathway I described earlier. That's calcium signaling. They end up raising calcium within the adrenal cells. This leads to increases in the aldosterone synthase and a protein called STAR that regulates cholesterol within the adrenal. And that leads to autonomous, renin-independent aldosterone production. So a point important for the clinicians in the group to think about, and that is how common is primary aldosteronism. And if you reach into the literature, depending on how far you go back, you're going to see all kinds of numbers that indicate potential prevalences of primary aldosteronism, everywhere from 2% to 20%. And to get at that, I wanted to show you some recent data published by a collaborator, friend, Anand Vaidya, at Brigham and Women's University. And what he's shown recently using urinary cortisol, urinary aldosterone excretion here, and this is the number of participants in the bottom, and you can pull up his article, it's an amazing article, but he looked at untreated normal tenses and found actually that 9% meet the current criteria for aldosterone excess in the population under a sodium suppression diet, which is what these individuals were on. If we then look at stage 1 hypertensive patients, the number jumps to almost 16%. We look at untreated stage 2 hypertensives, we go up to 21%. And then finally, they kept their resistant hypertensives under treatments, and they found that 24%, quarter of patients that are resistant to drug therapy, hypertension-wise, have inappropriate aldosterone production. So his findings in general suggest that PA is a disease continuum, and that renin-independent aldosterone production is even seen in normotensive individuals, and that the conventional aldosterone-renin ratio that's taken with spot blood measures may not be sufficient to detect all PA cases. Their work also suggests that under suppressed renin conditions, high sodium diets, that serum aldosterone levels even lower than 10 nanograms per deciliter are not necessarily normal or low. And finally, their work is pushing the concept that a 24-hour urinary aldosterone is superior to a spot blood level and helps in the detection. And again, from a prevalence point, even normotensives have some cases of inappropriate aldosterone, and that 10% of adults may be affected. And that sort of sets up the premise for the work I'll talk about today. And that is that autonomous aldosterone production is very common and represents a continuum. And therefore, we hypothesize that we could detect the early intraadrenal histopathology of a disease process in normal adrenals. And specifically using immunohistochemistry for that enzyme that I've spoke about already, the aldosterone synthase, that's so closely and tightly regulated by angiotensin II and would be regulated inappropriately by mutations that cause aldosterone excess. So I'm going to talk about 4 studies total, but 3 are very similar, done by actually 3 Japanese fellows that have worked through the University of Michigan doing these studies. And the first involves characterization of an abnormal structure of cells that have been called aldosterone producing cell clusters by my fellow Nishimoto. And the second looks at these structures as we age, using a large cohort of normal adrenals. And finally, a study by Dr. Omota, where he looked at aldosterone driver mutations in these aldosterone-producing cell structures. And this was all made possible by some technologic advances that came about over the last 10 years as well, that allow us to study RNA and DNA in FFPE tissue blocks. And by doing that, it opens up studies, your studies, our studies, to a large pathology archival tissue collections. And it also allows us to do immunohistochemistry to target the cells that we capture. And therefore, we work with serial sections from adrenal tumors. We do the immunohistochemistry for CYP11B2, and then we are able to capture selectively the regions that express the CYP11B2. And then we can analyze and do DNA sequencing with a system that was also available when we came here to the University of Michigan. And that's a deep sequencing method, targeted deep sequencing using ion torque technology, which gives us 1,000 to 2,000x coverage and allows us to sequence for mutations in 5 nanograms of degraded DNA material. So the first study ran by Dr. Nishimoto studied these structures here, and he coined the term aldosterone producing cell cluster for these aldosterone synthase expressing clusters of cells that didn't look like the normal glomerulosa. And recently it's been suggested by a pathology consortium that this terminology be shifted to aldosterone producing micronodules, but today I'll be calling them APCC. So Dr. Nishimoto was able to go in and capture by laser capture and by visual capture these structures to a degree that we could get RNA and we could do whole transcriptome analysis. This is a PCA plot which separates based on transcriptome differences, and we can see the APCC cluster closely with zona glomerulosa cell RNA, and whereas the fasciculata and the reticularis separate and form distinct patterns of gene expression. If we look at a microarray heat map at the genes that are the highest in the APCC versus the other zones, we see the #1 gene difference, which shouldn't be a surprise because we used it to target the structures, is indeed the aldosterone synthase CYP11B2. And if we quantify that with quantitative PCR, we see that there's much higher levels of mRNA for aldosterone synthase in these structures. So my take-home message on the APCC is that they're aldosterone producing foci of cells just in the glomerulosa region, but their cells extend on into the fasciculata. They're similar to glomerulosa cells, but with even a greater capacity to make aldosterone. So study 2 looked at a large Michigan cohort of normal adrenals, and this was ran by Kazunaba, and he found that APCC associated with aging. And this is the classic vision we think of when we think of aldosterone producing cells. This is an adrenal gland stained for the aldosterone synthase. And what you see is the aldosterone synthase forming a tight pattern just under the capsule that we would historically have called the glomerulosa. And if we look at that in H&E, we see a clear glomerulosa, separate fasciculata. And if we stain and look at the higher magnification of CYP11B2, you can see a nice glomerulosa that's positive and a fasciculata that's negative for this enzyme. But what he found with aging was a distinct change in the zona glomerulosa, where we started to have a discontinuous glomerulosa with the development of APCC. Here we no longer see a distinct glomerulosa, but we see a buildup of structures like this, which we can look at and see under immunohistochemistry quite well. These can be quantified, and what Kazu showed is that we have a nice continuous glomerulosa as the predominant form of adrenal in early ages, but these change with age as we go more and more down this path of discontinuous glomerulosa. And that associates quite well with an increase in the aldosterone producing cell cluster area. So is this an associated change in aldosterone dysregulation? And for that, I relied on our collaborators at Harvard, Dr. Rainey, and what he found using a modulated physiology of human subjects, putting on the same patients on a high sodium and then a restricted sodium, something he called the SASI. He was able to demonstrate that inappropriate aldosterone production does increase over age. And this can be shown in a number of papers where he's using this technology. We followed that with a very tight analysis of a Japanese cohort of adrenals, normal adrenals, from individuals who had no hypertension in their history. And we found even in these normal individuals that APCC did accumulate with age. And then in this case, Dr. Amata went in and isolated every one of the APCC in these over 100 cases. And what he demonstrated were though the aldosterone driver mutations found in tumors were also found in these APCC structures, tumors that normally were driven by L-type calcium channel mutations, ATP channels, etc. So again, all of these studies agreed that APCC are common in normal adrenals, and many have driver mutations that are found in tumors. But the question was, do APC progress and cause disease? And that's a difficult thing to get at using human tissue, but we were able to try to address that by study 4, which was looking at the cellular and genetic causes in bilateral disease. Again, bilateral disease is the most common cause of primary aldosteronism. And to do this, we were able to get 15 very rare resected adrenals from bilateral disease patients. And we carried them through the same protocol with IHC and capture. But immediately we saw that these adrenals from these primary aldosteronism bilateral patients had a buildup of these APCC structures. And in this case, Dr. Amata was able to capture those and study them. First, he just looked at some calculations of APCC area and showed that it increased significantly in IHA patients compared to normal controls. So here we have the normal 53 controls, most of them laying right across the 0 line, but as I showed you earlier, some have APCC. Here we see the IHA, N equal 15 adrenals again, and we see that there's a 6-fold increase in APCC number in the IHA adrenals. If we look specifically at area, we also see there's an increase up to 0.25 millimeters versus 0.16 in age-matched controls. So we're increasing both size and number. Finally, again, Amata went in and isolated all of these APCC structures and did mutation analysis. And when he did so, this is the IHA mutation analysis profile. And what is striking here is the majority have mutations in this CAGNAT1D. This is an L-type calcium channel that regulates calcium homeostasis in adrenal cells. And it was a very different pattern than in a race-matched group of aldosterone-producing adenomas, where casein J5, a potassium channel, is the predominant cause. So this tells us that in bilateral disease patients, we see somatic mutations that cause tumors and cause autonomous aldosterone production in tumors. We see mutations are in the CYP11B2 positive foci and not in the normal adjacent tissue. And these findings suggest to us that somatic mutations are not only a cause of adenomas in this disease, but also in bilateral primary aldosteronism. And of note for possible clinical therapy, because most of the mutations were found in this L-type calcium channel, there is the potential of an add-on therapy of using calcium channel blockers as an IHA therapeutic option. So in summary, this is our working hypothesis for the progression of aldosterone disease within the adrenal. We start out with adrenals that are devoid of driving somatic mutations that cause aldosterone, but over time, we see that there's an age-associated appearance of clonally derived APCC with somatic mutations, as we can see in this cartoon here, and that some of these over aging can progress onto tumors, whereas they can also just increase in size or number, leading to clinically relevant either bilateral or unilateral disease-causing cells. And this goes along with a progression that Anand presents, where we go from clinically relevant renin-independent aldosterone production in normal tensives, and that moves into our classical biochemical cutoffs for PA, and then finally to the resistant hypertensives that we see here with hypokalemia. So with that, I'll stop. I just want to mention that, again, we're part of a rather large interest group for primary aldosteronism and adrenal research in general. These are our research teams. These are our clinical teams. And I really appreciate the hard work of the fellows who did the work I spoke about today, Kosh, Kazu, and Kay. And then again, we have a very robust clinical and basic collaborations internationally as well, particularly with the Japanese collaborators at Tohoku University, Dr. Sato and Sasano, who provided fellows and samples for this study. And with that, I'll stop and be glad to take questions if there are any.
Thank you, sir. I think it was a brilliant talk, and thank you for finishing right on time. Can we have some comments from a chairperson?
Yeah.
Dr. Kotwal, sir? Would you like to? Sorry.
Dr. William Rainey, it was a very nice overview of the primary aldosteronism. As you told us that the primary aldosteronism basically is a continuum from the normotensive individual to resistant hypertension, and there's a renal independent aldosterone production. My question to you is that even 10% of the normotensive individuals have renal independent aldosterone production. Is there a case in point to treat these individuals?
You know, this is a extremely, at the moment, a fascinating question, of course, and warrants more research. Anand's work on this, he's got the largest study showing that normotensives do have inappropriate aldosterone production. And some of his studies are showing that these patients are the ones that are going to progress to having hypertension later on. So what do you do with these patients? I don't know. And I'm not sure he's going to push that they're treated yet, but they are the patients that are going to progress. And therefore, if you picked up a normotensive that had inappropriate aldosterone levels, these would want to be patients that you have revisit and monitor as the disease progresses.
Thank you.
Dr. Sanjay, sir, you want to add something? Yes,
Professor William, the very lucid talk. I have a question like the Yano memo, that tribe which you have studied, the sodium level is very, what was their serum sodium level?
It's normal. If you go back and look at these papers from the 70s, it's fascinating that they have a completely normal potassium and sodium level. Even under severe stresses, the second article that I mentioned followed women that became pregnant. And again, maintained this extremely low sodium diet, but also maintained normal physiology. So it's a fascinating group. Interestingly, and there's a lot of debate about whether this is because of the genetics of the tribe itself, but there is no hypertension in that tribe, even as you age.
Okay. So the other question is that are you planning to have change the hypertension guideline, treatment to hypertension guideline, that you treat the patient with calcium channel blocker plus this what you call aldosterone inhibitors, the combination to treat most of the hypertension?
Well, we're doing I'm doing this in collaboration with my clinical collaborator, Anand at Harvard. So we're actually doing a pilot study of bilateral hyperplasia or bilateral aldosterone excess patients using calcium channel blockers to get at that point. is it a useful treatment? There's also other populations where calcium channels are common in tumors. We've just published a study recently in African Americans where calcium channels are the primary cause of disease in men. I don't think there's been a study yet of patients in South Asia to determine what the prevalence of mutations are. It'd be interesting to know. But right now, those studies are being done, and I think that we have to see what the outcomes are before a non makes a guideline. I'll just be riding along somewhere.
Thank you.
Dr. Vivian, ma'am, you have anything to add to this?
Well, thank you very much. This was actually a wonderful, lovely, fantastic continuum from normotensive all the way to severe hypertension and the basic science behind. Now, as you know, I was just wondering, just slightly digressing, the holy grail to primary aldosteronism for a lot of us clinicians is the distinction between bilateral disease and autonomous primary aldosterone producing adenomas. Now, in any of your lab and your data, do you think any of your genetics could help us translate this and distinguish these two spectrums?
Yeah, my lab has a rather diverse interest. In the steroid side, the University of Michigan, Tohoku University, also Queensland, Australia, we've all been working on, and as well as a couple labs in Germany, have been working on circulating biomarkers that would differentiate unilateral from bilateral disease. And it's been very complicated because it looks like, depending on, there's distinct race differences in the mutation prevalence. And now, if we look at the Japanese population, it looks like there's a very good biomarker, steroid biomarker, called 18-oxocortisol that will differentiate the patients that have unilateral disease, the bulk of them, from patients who have bilateral. And that's because the major genetic cause of tumors in Japanese is KCNJ5 mutation, about almost 85%. And those tumors produce an unusual cortisol compound. And so it's been very useful for differentiation. So we're also starting to work on circulating DNA to see if we can pick up mutation status that might help. But that steroid biomarker right now is the most promising. It just doesn't work as well in European populations where KCNJ5 tumors it's about 45% of the tumors are caused by KCNJ5. So in that regard, it's very important to know the mutation prevalence within the population of individuals you'll be working with.